Hi, everyone. Arsalan Arif here with Endpoints News, and thanks for joining us today for Identify and Quantify Point Mutations in Circulating Tumor DNA, the ddPCR approach. We're sponsored by Agilex Biolabs, and please welcome our guest, Dr. Kurt Sales, Chief Scientific Officer of Agilex Biolabs. Kurt has over 25 years of experience in his career, spanning research and development, early drug discovery, and regulated bioanalysis across preclinical and clinical drug development. At Agilex Biolabs, Kurt is responsible for scientific integrity and strategic research and development. If you have any questions, be sure to hit the Q&A button at the bottom of your screen. We've reserved some time at the end of this webinar to answer your questions. Now I'm going to pass it over to Kurt to get started. Kurt?
Thank you very much, Arslan, and welcome back, viewers, to this webinar series. So today's webinar really focuses on digital PCR. It really has redefined molecular biology research, and this webinar really is a little bit of a snapshot on how we can use digital PCR in our clinical settings, specifically for clinical trials in molecular research and drug discovery. So by way of introduction, I'll provide a general overview of the landscape, the drug development landscape and the clinical ecosystem, which really just frames bringing new biological and chemical entities to Australia to conduct clinical research, an overview of Agilex Biolabs, what we do, how we measure things in a clinical trial, and then we'll launch into the webinar for today and a brief case study. So the webinar really is focused on ctDNA and cancer. And cancer is one of the leading causes of mortality and morbidity worldwide. And over the last decade, there have been massive advances in personalized medicine, thanks to genomics and proteomics. And precision oncology in particular is an emergent area of focus, and the use of circulating tumor material in the selection of intervention therapies and stratified medicine is of enormous interest in recent years. But developing a new medicine for a therapeutic indication is a stepwise process, and it takes several years, aided by discoveries such as the digital PCR. So drug discovery generally starts with identification of a biochemical target, and this has been really come to the fore using gene sequencing or genomics to find differentially regulated target genes associated with a disease or indication, or the utilization of proteomics to find protein targets that are differentially regulated either up or down within a cell or as cellular surface, and which are also thought to play a role in disease progression or disease regulation. So the target might be a cell surface receptor, it might be an intracellular signal transduction pathway or intracellular protein or intracellular target gene or mRNA. But more recently, the exosome and the concept of liquid biopsy has reshaped the oncology space, particularly in personalized medicine. So what the liquid biopsy does is it provides an instantaneous snapshot of what we call the tumor circulome, the subset of tumor-derived factors that circulate within the bloodstream and can be used as a source of tumor markers. These originate from the tumor, and they include proteins, which are differentially regulated, and also circulating tumor DNA. So this slide provides a brief snapshot of the development landscape, right through from early drug discovery, where you're identifying those targets that are so specific for the disease indication, you're optimizing your leads, and you're generating your first molecules to take through into preclinical and clinical safety studies. And these take up to 15 years, as you can see from the time scale at the bottom. And the period that we're really interested in here is the bioanalytical method development, validation, and sample analysis. And that's where Agilex Biolabs sits and the topic of today's discussion. So we fit within the clinical ecosystem. It comprises multiple stakeholders from labs to CROs, hospitals, and phase 1 units within which the clinical trials take place. And we are just one provider of many that forms part of this clinical ecosystem within Australia to enable fast turnaround and rapid development of new drug entities. So we are the leading bioanalytical laboratory. We've been around for over 25 years. And our main service lines span from preclinical toxicology in multiple species to provide safety studies, through to clinical studies. And overarchingly, we specialize in regulated bioanalysis, either through liquid chromatography or mass spectrometry for small molecule bioanalysis, or large molecule bioanalysis by ligand binding assays, immunobiology analysis to really provide a more detailed snapshot of what the drug is doing in terms of pharmacodynamics, and that includes evaluation of immune markers using flow cytometry, and then also differentially regulated genes or microRNAs or mRNAs, which we use our sensitive digital PCR suite. We also provide vaccine support and then overarchingly throughout the company biomarkers, either as an exploratory endpoint or as a regulatory secondary endpoint. We're the only FDA inspected laboratory, and we've been around for over 25 years and accredited by the National Australian Testing Authorities for ISO 17085 and also OECD GLP. So what do we measure in a clinical trial? Well, multiple different parameters. And I'll take you through the slide, which is quite busy, but really provides a high-level snapshot of what we generally do within a clinical situation. So if you imagine a patient or a subject in a clinical trial dosed with a drug in the top center, the subject will undergo blood sampling at regular intervals, and the blood is then either separated into serum or plasma for pharmacokinetics, immunogenicity, and biomarker analysis, either by tandem mass spectrometry or ligand binding assay by immunoassay, or we can isolate PBMCs, which is the white blood cell, from the blood, and we can culture these, we can stimulate these ex vivo to provide cytokine release assays and powerful information, which really gives us an idea of what the drug has actually done to the patient's cell. And we can measure using our suite of images, and such as the Luminex Magpix or the MSD QuickPlex SQ120, multiple different parameters in terms of biomarkers. We can tag these cells with fluorescent labels, antibodies, and we can measure changes within the cell or changes on the cell surface by flow cytometry. And then in the right-hand side here, the concept of liquid biopsy. So that really is the whole blood, and the whole blood then contains a variety of circulating material, not only cells, but exosomes and ctDNA as well in those subjects that might have cancer. And so we can take the liquid biopsy or the blood, we can extract ctDNA from the liquid biopsy, and we can perform either PCR and digital PCR or quantitative real-time PCR. We can also subject that material to next generation sequencing, and this really provides information that is highly specific to that specific individual within the clinical setting. And this information gives us a snapshot of the patient's genome and the circulome and how they respond to a drug. And some patients might respond better to a therapeutic regimen than others due to some mutations within the ctDNA. So what is ctDNA? So circulating tumor DNA, it really is one of the most extensively characterized component of the tumor circulome, so the circulating tumor material. And really, it's the only component on which an FDA-approved companion diagnostic is based, and this was developed for non-small cell lung cancer. And I have put the reference at the bottom in case you're interested. But what really is circulating tumor DNA? Well, it's the fraction of cell-free circulating DNA that originates from the tumor. The mechanism by which it's secreted from the tumor isn't widely known, but in general, ctDNA consists of fragmented DNA from 80 to 100 base pairs in length. So regional studies identified mutations within KRAS DNA in colorectal and pancreatic patient samples, and that really brought ctDNA to the fore in terms of mutation detection and molecular biology. And it gives us really powerful quantitative or qualitative genomic information on the patient's disease state, profiling mutations, deletions, loci in ctDNA, really provides us with a powerful snapshot that is highly specific to that patient and can guide the generation of specific therapeutic regimens in stratified medicines and personalized medicines. And so it's really widely used for diagnosis of cancer, early diagnosis by, for example, identifying mutations within KRAF or B-Raf proto-oncogene or missense mutations within certain target genes. And this is very powerful as it can be used to stratify patients in a clinical trial, as those mutations for specific patients might lend them to respond better to certain treatments than subjects which might have a different mutation. And so this is reshaping the way that we're thinking of personalized medicine for oncology in particular. So we are using the BioRad Droplet Digital PCR suite. Consists of multiple different readers. So if you imagine you've got your sample, you've extracted your ctDNA, and you then prepare your ctDNA target with your master mix, and you then add that sample with a target to the automated droplet generator. So what this does is it partitions your sample randomly into 20,000 droplets. Each droplet is an individual PCR reaction. And then you add your droplets in 96-well plate to just a normal thermocycler. So we've got the BioRad CF96 Touch RT-PCR thermocycler. And the PCR reaction occurs as per normal up to cycle 40, which is the usual amplification strategy. And the PCR then amplification occurs within each droplet, and positive targets for fluorescence are then evaluated using the BioRad QF200 droplet reader. And then we use our software to determine concentration of targets using Poisson statistics. So to demonstrate the utility of the digital PCR system in a clinical trial situation, we have a short case study where we aim to identify and demonstrate the capacity of our BioRad QX1000 droplet digital system to detect BRAF V600E ctDNA. So just as a way of introduction, the BRAF proto-oncogene encodes a cytoplasmic serine threonine kinase with a key role in regulating mitogen activated protein kinase signal transduction pathways within cells. And then activating missense mutation codon 600 of exon 15, the V600E, has been identified within the B-Raf gene in multiple neoplasms, including melanoma, colorectal carcinoma, papillary carcinoma, and so on, as being crucial for the regulation of these diseases. And in this study, what we did was demonstrate the capacity of our BioRad system to detect BRAF V600E circulating tumor DNA as a measure of disease state, which could then be applied within a clinical trial situation to detect these mutations within a certain patient population. So at the bottom here, this is the missense mutation of thymidine within the B-Raf proto-oncogene. So how did we do this? So generally, samples are collected from subjects with tumors, and circulating tumor DNA is then extracted and using a commercially available kit from QIAGEN, and then stored. And using a commercially available hydrolysis probe assay to amplify either the V600E or the wild type, using either wild type labeled with a hex fluorophore or with the 5-hydroxyfluorosine for the mutation. And so we added our template ctDNA into the PCR reaction, applied it into the droplet generator. It got partitioned into 20,000 individual droplets, This was then subjected to a PCR amplification, and then the partition PCR reaction analyzed on the QX200 droplet reader. So then the data was analyzed using QX Manager. We've got the regulatory addition fully under change control and validated. And the QX Manager then uses Poisson statistics to identify and calculate the fractional abundance of the circulating tumor DNA versus the wild type. So one of the first things that that we did when optimizing this assay was to actually look at the droplet generator and the size of the droplets. So we've got a little bit of a time-lapse movie here, which shows that using the automated droplet generator function, we get really uniform droplet size within a good range, quite a narrow pixel range. So that was the first thing that we did. And then we went on to evaluate the difference between the standard qPCR and the digital PCR setup. So as I mentioned, the thermocycler is utilized for both. The amplification step is exactly the same. The main difference between qPCR and digital PCR is that in qPCR, you have one sample in a tube, whereas in digital PCR, you've partitioned the sample into 20,000 droplets. And each droplet essentially is equivalent to a tube. So you have the power of 20,000 versus the power of 1. So that's the first difference. The second difference is if you imagine this is your PCR amplification, you've got 40 cycles that you're amplifying your target over, and over 40 cycles you're generating relative fluorescent intensity as that amplicon becomes amplified up. And so as you can see, for those really low abundance, so ctDNA is very low abundance targets, we only start seeing an increase in fluorescence around about cycle 35 and above, which is close to the negative control, close to the water blank, and you cannot confidently provide a good analysis, at such a late CT. And so generally what you're looking at is your change in Y over change in X to identify what the cycle threshold is and calculate your abundance in that way. And so for an assay of this nature, qPCR isn't really sensitive enough to be able to do on its own. And so you then move to the digital setup, which is different insofar as the reaction is taken to completion, so up to 40 cycles. And what you have is really a binary readout. So all droplets are analyzed by the reader and are analyzed to either be positive fluorescence or negative. And so those samples that are positive for fluorescence get calculated, and the software applies it Poisson statistics to then derive a copy number. And so you can see, using the digital setup here, we were able to determine that there were 13 copies in a reaction for the mutant and 8 copies in a reaction for the wild type. So it's very, very powerful in being able to accurately quantify ctDNA. In terms of the digital setup, whereas the qPCR really resulted in CT values well beyond the reliable range of 35 cycles and above. We then determined the limit of blank. So the limit of blank is really a 95% probability that a sample containing no target in multiple replicates gives a consistent value. So consistently, we were able to determine that the negative control, we would consistently derive 0 fluorescence, which to be expected. And then within that 5% false probability, the 95% confidence interval, we could determine the lowest limit that we could reliably say was a positive reaction. And so we determined that to be 3.5 copies for the V600E and 1.2 copies for the wild type. So the next thing that we went in and did was to have a look at linearity. So we took our sample and we serially diluted it from neat right down to 1 in 64. And so what you can see from this top graph is a really good linear range from neat right down to 1 in 4. But then it started tapering off after a 4-fold dilution. And became less sensitive. And the fractional abundance started wobbling also around about 4-fold dilution. So how do we correct for this and how do we improve the sensitivity of detection so that you can dilute out your sample and really get to an absolute copy number? So because the reaction is partitioned into 20,000 droplets, for a digital PCR setup, you can actually do the reaction in singlet because you've got the power of 20,000 within one well. And so the way of improving on the dilution factor is to increase the data set. And by increasing the data set, to increase the number of replicates, you can then tighten down the fractional abundance. So that's what we did, we did the sample down to 8-fold dilution, as you can see here, and you can, from this graph, observe that the fractional abundance really is impacted at that lower dilution. But the minute we actually increase the number of replicates, we can then increase the confidence right down. So we can have as much as a 62% increase in the confidence interval ratio and the fractional abundance, and increase the sensitivity of the assay by doing more replicates. So that was a powerful observation in how you can change your experimental design to really tighten down the data set at that lower end of the curve. We then investigated our inter and intra-operator accuracy and precision. And so we used the digital droplet generator mode for this so that we could standardize droplet generation between our 3 operators so that we were only looking at the inter and intra operator accuracy and precision. And so what you can first observe is that the intra-operator precision, we could easily get CVs of less than 10%, so 8.7 of operator 1, 4.5 for operator 2, and 4.9 for operator 3. And then with inter-operator precision between the 3 operators, we could get really good consensus, so about a percentage CV of 3.3%. So incredibly reliable, incredibly consistent, and very, very accurate in terms of being able to measure the ctDNA in this particular assay. We investigated freeze-thaw stability. So one of the issues that you often encounter with DNA and RNA particularly is that they're quite prone to shearing stresses when you freeze them down. And this can really impact the amplification of the target of interest. And so what we did was we took the sample down to 5 freeze thaws and did the PCR reaction at each freeze thaw, and I've only put sort of 1 freeze thaw versus 5 on the slide. And what you can immediately see, because we're doing the digital PCR where the reaction is taken to completion, and it's a sort of a binary fluorescence versus no fluorescence that the software is evaluating, you can see from this slide that you get really good measure of fractional abundance for, and this is the mutant only, between 1 and 5 free stores. So you do get a little bit of a difference after 5 free stores, but you can still reliably detect the differences that you need to using the digital setup. So really really sensitive over and above a traditional PCR assay. The last thing that we did was to really have a look at the post-PCR droplet stability. So sometimes you might want to come back to your PCR. Once you've done it, you might want to repeat it. Something might happen, electricity might go off, the reader might give up. There are often things that can go wrong in a laboratory situation, and you need to know what level of stability you have in your sample, once you've actually either put it through the PCR reaction or you've partitioned it using the droplet generator. So what we did was we generated our droplets and we left them in the fridge, and we analyzed our targets at day 0, so on the day of preparation, and then day 7, day 14, and day 28. And so the first thing that you can notice is that the droplets actually remain quite stable over time in terms of the actual droplet count. There is a notable and slight increase with the droplets over time, and this could just be down to the way that they behave at 4 degrees, but reliably we were able to detect similar differences between the wild type and the mutant in terms of copies per microliter, very consistently in terms of the fold difference over the 28-day period. So the fluorescence remains stable for up to a month, and you can accurately detect those fractional abundance measurements. So really, this method is not only robust in terms of the accuracy and precision, but also it's robust to interruptions in terms of the assay, which in a laboratory situation is crucial for being able to have consistent and reliable data. So I hope that this webinar has given just a brief snapshot of how we can apply the digital methodology to something such as ctDNA in a clinical trial setting. So we could measure pharmacokinetics, we can measure pharmacodynamics associated with target genes, and these could either be mRNA or they can be DNA, and we can utilize the same premise to be able to amplify up any target to be able to determine whether the drug has elicited a reaction which is warranted, or whether there's an off-target or a differential regulation of a pathway that might be influenced by the therapeutic strategy. So digital PCR allows us to detect tumor-specific mutations within circulating tumor DNA. It's more sensitive than traditional PCR and allows absolute quantitation of copy number. And the accuracy and precision is phenomenal. It's really superior, and that's really down to the partitioning into 20,000 droplets and the power of the 20,000. So you're counting large number of molecules individually in the digital reaction versus a single reaction for a traditional PCR. And so the take-home message really is that digital PCR enables precise and highly sensitive quantitation of nucleic acid, which really, as I mentioned, in a clinical trial setting is crucial and opens the door to personalized medicine and better outcome for patients. And on that note, I will thank you for your attention and happy to take any questions.
All right, Kurt, thank you so much. And some of my questions over here might be reiterations of what you've just concluded over here, but and thank you for your presentation. But tell us, Kurt, what are the advantages of digital PCR for measuring circulating DNA, and why is that important? ctDNA, and why is that important?
So the advantages really are it's the power of the digital PCR, and that arises from evaluating individual molecules as positive or negative for the parameter, But because you partitioned that sample into 20,000 droplets, you have the power of 20,000 mini PCR reactions within a tube versus a traditional PCR reaction where you are only evaluating a single reaction. And so for many assays, the sensitivity of the digital PCR is significantly higher than traditional PCR analysis. The accuracy and precision of the assay improves by counting those larger numbers of molecules individually. And so really the sensitivity of the digital PCR will facilitate detection limits that really redefines our understanding of disease onset, progression, and reoccurrence, specifically within a clinical situation such as the one that we deal with on a daily basis at Agilex Biolabs.
Okay. Now tell us what are the advantages of digital over conventional PCR?
It's definitely the sensitivity. So, PCR, when it first emerged, it really changed life science research and molecular diagnostics, decades ago. And so digital is now changing that field even further by being able to push the limits of sensitivity. So because there's an endpoint analysis we're dealing with sensitive algorithms within the software, we can actually detect those really low copy numbers and low fractional abundances of things such as mutated genes. And that really is crucial for those early diagnoses of cancer, for example, where the tumor might be small and just emerging, it might be hidden somewhere, and we can use a liquid biopsy to be able to determine whether there's a likelihood that someone might develop a cancer or whether there's a tumor that is shedding some of its material.
Okay, very good. And now lastly, how, in your view, will digital PCR revolutionize personalized medicine?
So because of the sensitivity that we're dealing with, if you imagine a patient that has an unmet need, there might not be an appropriate therapy, or it might be a cancer patient that is either terminal or has gone through all of the known medications without success. What digital does is it allows us to stratify those patients, identify those key mutations that might be highly specific only to that patient, and we might be able to then find appropriate targeted therapeutic intervention strategies for that specific patient. So I think the revolution is around being able to prepare those libraries for next generation sequencing, and then being able to use the digital to confirm those molecular targets, and then develop appropriate therapeutic strategies towards those targets, and being able to then provide an outcome for patients that might have a specific mutation.
All right. It's always a pleasure to have you here, Dr. Kurt Sales. It's all the time that we have for today. I want to thank everyone in the audience for tuning in. And again, Kurt Sales, thank you for sharing your time and expertise. We also appreciate Agilex Biolabs for sponsoring the discussion, as always, at Endpoints webinars. If you'd like to rewatch a webinar or share it with a colleague, a link for on-demand viewing will be sent tomorrow. I'm Arslan Arif for Endpoints webinars. Thanks for joining us, and we hope to see you at a future event. Thanks again.
Thank you very much, Arslan. Thank you, viewers.